Hi, thank you for joining me in this episode of GradSWE UCLA presents Women in Science, We Have the Power to Change the World. My name is Yifan Gao, a PhD candidate from Civil and Environmental Engineering Department at UCLA. I'm also the International Student Event Officer at GradSWE UCLA. GradSWE stands for the Graduate Student Community of Society of Women Engineers. Our goal is to provide support to graduate students and help them shine in their future career. We are going to host a series of interviews to female scientists, and we hope we can encourage more women to join the community of STEM. Our guest today is Dr. Georgia Squyres. She's currently a postdoc belonging to Biology and Biological Engineering departments in the Newman Lab at Caltech. Her area of interest is how bacterial cells coordinate large-scale, spatial-temporally organized processes during biofilm development. Hi, everyone. I'm with Georgia today at Caltech. So it's my first time doing it in person, and thank you, Georgia, for being my guest.
Of course.
For the interview. So should we start with some introduction?
Sure.
Yeah.
So my name is Georgia Squyres. Right now I'm a postdoc with Diane Newman here at Caltech. Just a brief, I guess, scientific biography. So when I was young, I was always interested in math. And so I started out in undergraduate as a physics major at Columbia in New York City. About halfway through, I had a minor switch and moved to biophysics.
It's
I fell in love
switch?
Minor switch.
Okay.
I fell in love with biology.
Yeah, you said it.
I could tell the story if you're interested. And so I ended up graduating with a biophysics major. I worked for a year as a technician, learning some single molecule biophysics-y stuff. Then I went on to do a PhD in the MCB department at Harvard. And there I was doing something that was sort of half single molecule biophysics and half bacterial cell biology, trying to understand the molecular and sort of biophysical mechanisms of cell division in bacteria. And now, always for a PhD. And now for a postdoc, I've come here to Caltech to work with Diane to study biofilm development. So how individual bacterial cells inside biofilms coordinate their behavior in space and time. And so this sort of combines, first of all, a career-long passion for microbiology. I studied bacteria my whole career, really. You
abandoned physics then? Like completely, or like bio? I know you're the biophysics, but
I mean, there's still some of that. There's a little bit of mathematical modeling, for example, in my project that's been quite helpful. Maybe more as things go on, we'll see. And a lot of microscopy, so a lot of the single molecule work I was doing before is optics related, and so those skills have come in handy.
That's cool.
Trying to understand, a microbial lifestyle that's more directly relevant to the clinic and the environment, and that's also just more complicated.
Okay, I got it.
Sort of appealing. And so here I am.
Okay.
I've been at Caltech for almost 3 years. So next steps are on the horizon. I'm not quite yet. But, I'm sort of mid postdoc, starting to think about what's next.
So it's like in biology, like the regular postdoc time is 6 years, or
it really depends. Every subfield, as I'm learning, every subfield of biology is different. It just depends. I think for me, it'll take as long as it takes. I'm hoping like a 4-year timeline is maybe a sweet spot, but I mean, you never know. One of the things that's nice about these positions is that you have some flexibility to work with your advisor.
That's true.
As long as you have some funding and some momentum scientifically, I think there's flexibility, which is maybe a unique privilege to this type of career, which I appreciate. The way that I'm thinking about this postdoc, so when I'm finishing my PhD, I thought first about what I wanted to do for a lab. And then I sort of worked backwards to make a checklist of all the stuff I would need to get done as a postdoc to make that feasible. Both so that I could give a convincing job talk, so show that I had the training and the skills, but also like, what are the things that I need to set up? What are the things that are really risky that I should do as a postdoc when I have a little bit more
time,
freedom to take risks? What are the things that are so hard and crazy that I can't really ask somebody else to do them for me, but I should do them myself?
I got it.
Things like that.
So eventually you're gonna build up your own lab.
That's the goal.
That's the goal.
That's what I'm working toward. And so I have a sense of like, here are the things that I really need to get done as a postdoc. And once I've more or less worked through that list, that'll be when it's time to go.
Yeah, I'm looking forward to that. Yeah.
We'll see.
So yeah, so I think you wrote the article about something behind the nature because you published a Nature paper.
Nature microbiology.
Nature microbiology. And I read that. I know you have a sentence there, like if you are talking about your research, like cell division bacteria, like most of people's reaction is, oh, we already know that. It's all in the notebook. Like why you actually like wanted to dive into that deep for that? I know you're interested in microbiology, but why specific this one?
Oh, there's so many possible answers to that question. I'll give maybe a general one. So this is about really my PhD field, which is bacterial cell biology. I'm still trying to keep my connections to that field. I think there's connections with what I do now, but bacterial cell biology is like a cool, interesting field because there's a little bit of a dichotomy in terms of where the field is at. Famously, bacteria have really good genetics. And there's a way in which they're very simple. I'm like a lot of people that came from physics that I ended up in microbiology. To a physicist, a bacteria seems like a very minimal system for life, right? They have a smaller genome, there's just one cell. And so, you know, by analogy to physics, which is very reductionist, especially when you're learning, it's like, okay, I'll study the simplest cell that I can possibly find. Like the bacteria. E. coli, right? E. coli,
But so, I know like there's E. coli is not well studied.
It is and it's not. There's lots of funny things that we don't know. So, okay, so it's true that the genome is small. It's true that the genetics are good. And so there are some things that are uniquely well studied in bacteria for sure. However, the field that I was in, cell biology, a lot of the history of that field is driven by microscopy, specifically light microscopy. And bacteria are really small.
So unique.
Famously. So light microscopy in bacteria is just very challenging. First of all, just resolution-wise, the size of a bacteria is, I don't know, if you generalize it as a sphere in a physicsy way, like maybe a micron sphere, maybe a little less, the resolution limit of light microscopy fundamentally is around 250 nanometers. So you're very close to the limit of what the instruments can do. And because bacteria are small, the signals that you're dealing with are lower, etc. And so that field of imaging in bacteria has sort of lagged behind just for technical reasons. And so there are some things that we know better in eukaryotes than in bacteria.
That's definitely.
It's specifically like because of imaging considerations. And so the field that I was in is one of these types of fields. I was specifically in a lab that focused partially on the bacterial cytoskeleton. The term is a little bit of a misnomer. There isn't like a cytoskeleton in bacteria in the same way, but you know, thinking about filaments,
filaments, membranes, etc. They have something. It's not just
You know, the cytoskeleton doesn't give the cell its shape in bacteria. It's a very different thing, but they do have like homologs of actin and tubulin that make filaments that do stuff. And we understand sort of less about what these filaments do in bacteria because they're really small filaments inside really small cells. So that was the general reason we don't know about it. The reason that I was interested is cytokinesis is a physical process. In order to divide a cell, again, we'll be physicsy in general as like a sphere. Right? This is the cell with the membrane.
You these two together.
You know, in order to separate this thing, you have to deform the membrane away from a sphere, the low energy conformation. You have to do mechanical work to form this membrane. And bacterial cells are pressurized. So it's even harder, right? They have a bunch of turgor pressure pushing out, resisting your pushing inward of the membrane.
So they don't want to divide.
Like you have to apply physical force.
So it's like giving birth.
Well, so in a sense that also uses mechanical work. You need something at the division complex that is able to do mechanical work. And usually the way this works is you have something that takes chemical energy to physical. That class of molecules in biology is called a molecular motor, right? You know these well.
Molecular motor.
So this is something like dynein, myosin, kinesin, many other molecular motors as well. But the class that you might think of are the ones that walk along a tubulin. In our cells, there's a lot we don't understand in eukaryotes about how cytokinesis works. I don't mean to say that it's a solved problem, but there's some logic there that makes sense. There's actin filaments and myosin motors. We know that this can make a contractile structure that generates mechanical work, and we know this because that's how your muscles work. The sarcomere in your muscle cell is actin-myosin.
So it's basically you are doing analogy right now. Like the cells is trying to, okay. So if we are talking in a little bit easier way, so
Yes.
it needs some energy for the cells to divide.
Yes.
And you wanted to study the simplest model, like in bacteria. And then, because even in the eukaryotic system, we don't know like how cell divide. Yeah.
That's true. Yeah.
Yeah. Am I
Yeah.
summarizing
So
right?
here's the
Yeah.
catch that's interesting. So
Yeah.
the thing that I just described, where you make a little
Yeah.
contractile ring with actin and myosin, that happens in eukaryotic cells.
Okay.
In bacterial cells, there's still a ring of filaments. There's both the actin and
Yeah.
tubulin there, but there are not molecular motors
Oh, okay.
of this kind. So
Okay.
myosin, dyne, and kinesin are actually not in bacteria as far as we know.
Yeah.
And so there's still a structure, it still constricts, but there's no motor there to drive the constriction.
So what's driving the cells?
That is the question.
Okay. Okay, that's the question.
So that is why I
Yeah.
was interested. It's this
Okay.
very tantalizing
Yeah.
sort of physics of biology
Okay.
question.
Okay.
That was very naturally appealing to me. And I had a single molecule background, and it was pretty amenable to study with some single molecule imaging techniques. I wanted to learn microscopy. I really didn't have any microscope background when I came into graduate school.
So you basically got the,
So it was sort
yeah.
of it checked a bunch of boxes for me in
Okay, got
terms
it,
of the
yeah.
project.
So eventually, like in your lab, you're gonna, like in your lab,
Yes.
like you're gonna do the microscopy, like it is one field, and then like single molecule, probably like combination of everything.
Maybe. So that's my PhD work. I'm
Okay.
mostly out of that now.
Yeah.
So I'm not studying, for example, the cytoskeleton anymore, at least
Yeah.
at the moment. My interest is really in single cell behavior inside biofilms.
Okay.
So to draw sort of a arc
Yeah,
from my
yeah.
PhD to my postdoc, my PhD was about really trying to understand how the behavior of individual proteins inside a bacterial cell
Yeah.
can orchestrate complicated processes.
Yeah, that's true.
So the cytokinetic ring is made up, it's for
Yeah,
bacteria,
yeah.
a big complex with a lot of proteins in it. And somehow, if you just have these proteins, and each protein follows like a couple simple rules, like I
Okay.
bind to this with this
Yeah, yeah.
affinity, and I have these kinetics of
You're basically like more
enzymes.
specific with the protein. Yeah.
Yeah, so you
Yeah.
can just take all of the behaviors that all
Yeah.
these proteins do and get this like emergent,
Okay.
cool behavior.
Okay, so bacteria is using like a whole, like an army of protein to help them to do like everything, like even like simple division or simple cell activity.
Yeah, I think if you think about how an army works,
Okay.
your analogy,
Okay.
the way that the army works is everybody works together to carry
Yeah,
out a goal,
yeah.
but that is organized by somebody who's in charge,
Okay.
right? Like there's
yeah.
somebody, I don't know that
Yeah,
much.
I
Let me not be more specific.
Okay.
But there's somebody saying like, all
Yeah.
right, all of you are going to go over here and do
Yeah.
this. All of you are going to go over here. And do this.
Yeah.
In biology, in this case, in a protein complex, that's not happening, right?
Okay.
Somehow all of these proteins are working together to do something, but nobody's in charge.
Okay.
And so every one of these proteins has a short, specific set of instructions. Its biochemical properties, right?
Okay, yeah. So
the
you already
affinity it
coded.
has, etc.
Yeah.
And so if you just put all these proteins together with all their properties into a box, the bacterial cytoplasm, shake them up,
Okay.
somehow you get emergence of this complex behavior. They form into a ring structure that's dynamic.
Wow. Wow.
So this is like an emergent properties or
Okay,
emergent
got it.
complexity.
Yeah.
And in my PhD, I was studying this, how proteins come together to create macromolecular behaviors.
Okay, yeah.
Now in my postdoc,
Okay.
I'm like one level up.
One level up. Okay.
So
So you were studying single cell. Right now it's
yes,
biofilm.
now I'm in biofilm.
So
now the
biofilm,
question is,
yeah.
Oh, sorry, go ahead.
Okay, so I'm just trying to like, what is biofilm? It's like a community of bacteria. They are, if you are thinking about so like you never wash your like water bottle, and there's like something like really gooey.
Yep.
They're biofilms. So
Yep, they're on your teeth, the dental plaques that you brush off your teeth twice a
Yeah.
day. Those are biofilms.
Yeah, I'm wearing Invisalign right now, so I guess there's like a lot of biofilm.
Probably, yeah.
Yeah.
They're
Yeah.
really ubiquitous. I mean,
Yeah.
most terrestrial microbial
Yeah.
life, bacteria
Yeah.
and microbes are in biofilms.
Yeah.
So they're everywhere.
Yeah.
They're all around us.
Yeah.
And they cause problems in the clinic as well. If you have an infection with the bacteria that makes a biofilm, that can become very hard to treat and hard to get rid of. So if you're in the hospital, you can get things with hospital-acquired infections, etc. The heart of what these things are is a bunch of bacteria living and working together. So maybe if you picture a bacterial cell, if you close your eyes, maybe you picture just one cell, maybe it's got a little flagella, and it's living all alone in like a big empty world, a big black box.
Is that possible? Probably.
Yeah, I mean, that's, if you're a single bacterial cell, that is kind of what the world looks like to you. And there's some stuff that's nice. They can swim around and go where they want to go, but they have very poor control over their environment.
Yeah, that's true. So
anything that they make is going to be lost to the milieu really quickly. If they live together in these communities, they can start to organize their behavior. They can start to create a little enclave that they can control. And because there's so many of them, they can also specialize their behavior. To do stuff they couldn't do.
Like a human society or so. I
know
there's a movie, I forgot the name, but it was like a whole kingdom is living in a dust.
Oh yeah, exactly.
The elephant, I forgot.
It's like that.
Yeah.
You can picture that exactly. So they are able to sort of work together and transform their environment and do stuff that they couldn't do otherwise.
Yeah.
And for us, this is relevant because they become, for example, very resistant to antibiotics that we'd like to use to get rid of them.
Yeah.
Also, if you look at how these biofilms develop in the lab, or even there's evidence, from nature and from the clinic, we see that they pattern their behavior.
Okay.
So specific bacteria and specific parts of the biofilm are doing specific things.
Okay. So you want to understand that.
Over and over.
Yeah.
I'd like to understand that. And it's the same idea, right? That these individual cells, nobody's telling them where to go or what to do. Somehow they're able to figure out from the information that they have available, where am I in the biofilm? What time is it in biofilm development? How do I use that information to make a choice so that I'm doing what I'm supposed to be doing?
Okay,
Very interested in this.
I think that one is it's a level on, but it's also like really connected to what you're doing.
Yeah, it's also an emergent properties problem. Like, how do these biofilm scale phenomena emerge from single cell behavior? So I've gone up one spatial scale.
Yeah,
and the question is basically,
I got it. It's using the same technology to study like bigger problem.
Yeah,
like
so I'm also using microscopy.
Yeah.
By analogy, now I'm doing single cell imaging.
Yeah.
I don't have to do single molecule anymore because I'm tracking cell behavior, but I'm following individual cells of the biofilm and their dynamics and their behavior. So yeah, the logic is very simple.
Yeah, to be honest, I'm studying microbiology, but what I'm doing is I'm studying fungi or bacteria and using that to degrade contaminants. So I'm not looking at single cell level, but I think people should pay attention to the single cell level because like single cell will form the community and the community will do stuff. But if we don't understand like the single cell, we don't understand the community. We just know the results. I think
that's the pitch.
Yeah. So I really think it's super cool. And I think I asked you the question like why you got interested and everything, but I want to ask a few questions for my organization, like GradSWE, Society of Women Engineers. And like as a female scientist, I know you're studying microbiology. Like as a female scientist, like what challenges have you met in the way that you, for example, switching from physics to biology or like going into STEM? We just talk about, but you can talk to the audience.
There's so much to say about things truly are getting better and better for women in science. I really think that's true, and I think there's data to indicate that that's true. So the arc of history is on our side, but it's not easy. Definitely, there are times where things are hard. Science is hard, but it can be specifically hard to be a woman in science. I left physics, left physics. I mean, I still do a lot of physics in my research. Turns out you can have it all. I really switched into biophysics for research interest primarily, but I wasn't so sorry to come into biology for this reason either. There are more women in biology than in physics, and that helps a lot. I definitely felt the difference pretty much right away. And so throughout my career, I've really been trying to find places where there just are women to interact with
I got it,
as role models, as mentors, as peers, even as like trainees or mentees. I think it helps so much to have people around who understand your perspective. I think, again, this is something that everybody experiences, but maybe women experience a lot of. There are so many expectations placed on you as a young woman scientist.
Yeah, that's true,
And they're like fundamentally unwinnable. A lot of them are contradictory, and some of them, are maybe not consistent with who you are. Or who you want to be.
True,
And so the best thing I think that has helped me along the way is first of all finding other women,
Yeah.
like I said, who I can talk to and look up to, who can also help me identify, when something is an unfair expectation and when something is a challenging but fair expectation. Just comparing my experiences to other people's and getting some perspective, and also who can help me like embrace who I am and sort of let go any of the expectations that don't serve
You
me.
want to like talk to more women. I think because I'm engineering major. So I think STEM is definitely include engineering, and we don't have a lot of women or like even female professors. I think there's not a lot of like peers or women PIs I can talk to. So I think definitely if we can get like more women into this field, it's gonna be like better for our communication and also to have like a role model for our career also. Yeah, right?
Yeah. Definitely.
So we are talking about role model. Do you have any female role model?
Oh. Many. So my PI right now is a woman, which is fantastic. The first woman that I've worked for, and it makes as like a PI, it makes a huge difference. So obviously I look up to her example a lot, and I mean she's a phenomenal scientist, really bright, also just a wonderful mentor and lab head. So I learn a lot from her. Also people who I worked with as colleagues in the past. So there were some great women postdocs in Diane's lab who I learned a lot from as well. I like to think that in my scientific career, what I try to do anyway is like amalgamate all of the best traits of the people that I've trained under and trained with in the past. And so whenever I work with somebody, I try to like
to learn something.
Like, they do something cool. Like I'm gonna try to do that.
You're studying a bacteria is doing the same thing. Okay. I think we should, because as we said, it's not a lot of like female scientists around us. So like trying to learn from each of them is definitely really crucial for us to absorb more knowledge and also more like what should we do on our career, right?
Yeah. And I think it's also nice to see the diversity of women and gender expression and identities that can succeed in science. Not all of the women that I've worked with or trained with have been like me, certainly, but it's nice to see the many different approaches that people have that work.
Like equity, diversity is like really important in the scientific world. So my last question. Do you have any advice to our audience here? Like anyone who are thinking about pursuing STEM,
of course,
because not a lot of people, I think they have like inspiration or got encouragement from their, family or their background to use STEM major, especially female.
Do it. So I love this career. I love my job. I wake up every day excited to come into work. There's so much joy in being being a scientist, but especially a young scientist, especially a biologist. I'll make a pitch for biology. I think it's just a fantastic time to be a biologist. The technology is improving really quickly. And so it's a very open field right now to study anything that you're curious about. But truly, it's a field where you can follow your curiosity and your passion really far. And I would definitely do it.
Biology.
Science generally.
That's true.
That's the point. Is if you're somebody who is curious and you're motivated by curiosity, regardless of what science field you're in, that can drive you, and you're free to pursue those interests very far. My advice would be to find formal or informal mentors as quickly as you can.
Definitely.
My best advice for that is find somebody who's like one level up from you. So if you're an undergraduate, find a graduate student. If you're a grad student, find a postdoc.
Okay, that's really practical suggestion. I would say, I got There's like even there's like 1 year older than you, like they're senior and you are like sophomore or so, but you have like more experience, and some of them are really important.
And I think it's good to find somebody who is like what you want to be like. So somebody who you're like, wow, that person's a grad student and that's the coolest person I've ever met. They're so nice, they're so talented, they have so many skills that I think I would love to have. And then just ask them as much as you can about how they got where they are and try to do basically exactly that.
Okay. I think that's a really practical suggestion. Okay, So I think I'm gonna call them.
Sounds
Yeah.
good.
Okay. Thank you, Georgia.
Oh, what a
and
pleasure.
and hope you enjoy the video.
Good luck.
Bye.
Bye.
Thank you for joining me for today's episode of Women in Science. We have the power to change the world. Please hit the subscribe button and follow us to hear more stories from female scientists.